The Link Between the MSH2 Mutation and HNPCC by Kathleen Bostic, Micah Simone Howard, and Camden Davis. Next slide. Hereditary non-polyposis colorectal cancer, also known as HNPCC, is a hereditary disease with a high risk in cancer. It has been proven through research that many patients with such disease carry the MSH2 mutation, thus differing from other common cancers. Because MSH2 is in the MMR mechanism, deficiency in MMR increases mutation rate of colorectal cancer growth. This genetic mutation is also known as Lynch syndrome, as seen in the photo below. Next slide.
The mismatch repair mechanism is a mechanism that's able to repair a section of DNA that is damaged, and it happens due to a result of missense mutation, silent mutation, or nonsense mutation, or frame shift mutation. And so there are 5 steps within this mismatch repair mechanism. And the first step starts with 2 proteins called mut-S and mut-L. And these proteins are able to recognize the mismatched nucleotides and bind to it. The second step has to do with mut-S conjuring mut-H, and mut-H is an endonuclease, and this endonuclease cuts the DNA in close proximity to the error. And so once this occurs, an exonuclease will remove the error, and that will lead to a new strand being produced by DNA polymerase. And so lastly, in the 5th step, ligase will connect the new strand with the old strand, as you can see within the picture here provided. Next slide.
After analyzing our lab book and objective of this project, we were able to formulate the hypothesis that a missense mutation in the MSH2 gene will result in a higher probability of developing HNPCC. This because, as previously stated before, deficiency in MMR will ultimately increase mutation rate. Next slide. Features of the MRS413 plasmid. This is a plasmid with no insert, an empty vector. In this plasmid, one of its features are the AMP-R, which is the selection marker. It conveys resistance to the antibiotic ampicillin. HIS3 encodes for imidazolglycerol phosphate dehydratase, which is a protein. E1 is the origin of replication for PRS413. Ore's 2 micron allows the plasmid to persist with its host due to the partitioning systems and encoding of the copy number amplification. MCS is the multiple cloning site that the gene of interest gets inserted to after being digested with 2 restriction enzymes. And in the image as seen in the far right, it correlates to what I've just said. Next slide, please.
Okay, this was the method of how we did the plasmid extraction and digestion. The plasmid extraction and digestion method was used to isolate the plasmid DNA. Once the DNA was extracted, we used 2 restriction enzymes to digest it. Once digested, we could determine which plasmid DNA contained MSH2 insert through the use of agarose gel.
What you're looking at now is the result of what this experiment showed. And so all the way to the left, you can see gel image 1, and that has column 2, 3, and 4. And these 3 columns correspond to sample A, B, and C. In sample A, you can see that there is 1 fragment within the gel image, and the thickness of this fragment corresponds to 5,000 base pairs. And so once APE was used to digest both of these samples with XBA1 as well as APE1, we were able to correlate which plasmid corresponded within the digested enzymes. And so you can see in the green box that PRS RS413 has one fragment. And so that corresponds to sample A because sample A also has one fragment. In the red box, PMSH2, you can see that there are two fragments within the APE. And so that corresponds to column 3 and 4, which both have two fragments within the gel image. Now, one of the biggest details within this experiment was that sample B and sample C both could represent PMSH2 or PMSH2 prime. And the reason for this is because at this point in the experiment, we're unable to distinguish between either one of them because both of the genes generated by identical parents are the same after AP1 and XBA1 digestion. Next slide.
An additional method is the PCR and digestion method. Digestion is used to prepare DNA for analysis, thus resulting that digested is selectively amplified. Then next is to conduct a PCR reaction to amplify a mutation region of the MSH2 and digested samples of the PCR with the use of agarose gel electrophoresis. The use of restrictome enzyme BMRI is to distinguish the PCR products and ultimately identify which plasmid contains the muted insert. Next slide.
Here we have the results of the PCR and digestion. As you can see, sample A produced it, well, you can't see it, but it produced no product. Sample B was the wild type and it had one fragment of 900 BPs. Sample C digested was PMSH2 mutant, two fragments. One was 616 BPs and the other was 267 BPs. And sample C undigested, one fragment was 900 BPs. The yeast transformation method. We observed the transformation yeast strain AGY75 with mutant plasmid PMSH2 prime. The technique was accomplished by centrifuging the yeast cultures with supernatant the pellets and sterile water. Once finished, this method would measure the number of colonies on each plate. This was the yeast transformation result, and the AGY75 transformed with the PMSH2 prime with the following carried genes on media without uracil, tryptophan, and histidine. The growth pattern was growth. AGY75 untransformed with the following carried genes on media without uracil, tryptophan, and histidine. The growth pattern was no growth because it lacks the histidine gene. And the AGY75 transformed with PRS413 with the following carried genes on media without uracil, tryptophan, and histidine. The growth pattern was the growth. The fluoridic acid, known as the FOA method. This method was basically used to measure the mutation rate in the yeast strains with either the following genes from the previous lab. This technique works by resuspending the yeast culture until the specified densities were reached.
So in this slide, you can see the results of what this experiment produced. We had mutant 2, which was BMR1. And so you can see here that in the image to the right, those are where we were able to qualitatively observe bubbles within each of the colonies. And so below in the table, you can see that there are OD1, OD.1, and OD.01. And so these are all corresponding to what kind of bubbles the plasmids each had. And so in the AGY75 that carried the original plasmid of MSH2, there was no growth. And the reason for that is because PMSH2 has active MSH2, and that active MSH2 will decrease frame shift mutation, which will activate the GT16.5 URA3, which will thus activate the enzyme that encodes for that. And so this on FOA media will produce a certain toxin that can decrease the growth of the yeast. And so you can see that in all of the columns for PMSH2, there are no bubbles because there are no growth produced. In AGY75 that carry the PRS413, this particular plasmid does not have MSH2 within it, so it is inactive. And a result of MSH2 being inactive means that the frame shift mutation will increase. And when that increases, it will cause a lot of DNA damage. So the GT16.5 URA3 as well as the URA3 enzyme will be inactive. And that, in addition with the FOA on media, will not increase the toxin, it will decrease the toxin. And that will lead to a lot more mutation and thus a lot more higher growth. And so you can see that in the last column or in the last row, AGY75 that carries the PRS413 has the highest mutation rate. However, in PMSH2 prime, it actually has quite identical information. The MSH2 is also inactive because it is mutated, as well as the URA3 enzyme and the GT16.5. However, PMSH2 prime has a lower mutation rate in comparison to the PRS413, which is why you can see that there was only one bubble observed versus 4. So the mutation rate as a whole within this experiment was determined that PMSH2 had the lowest mutation rate, PMSH2 prime had a medium mutation rate, and PRS413 had the highest mutation rate. Next slide. So in conclusion, to bring you guys back to where we started, our central hypothesis was that a missense mutation in the MSH2 gene will result in a higher chance of developing HNPCC. So our lab mutation that we were given was within MSH2-H669Y. And within this mutation, we were able to subtract 11 base pairs that corresponded to a paper in 2007 written by GAMI. And so this corresponded to the mutation of MSH2H658Y, and this was located in the subunit. And within this paper, it provided a lot of information regarding that. The central conclusion of the paper was that when the mismatch repair mechanism is inactive, the MSH2 gene has a missense mutation, but it never gets repaired because the mismatch repair mechanism is inactive. And so because it never gets repaired, it results in a higher mutation rate. And this higher mutation rate is very common in a higher development of cancer. And so when the mismatch repair mechanism is inactive, it will result in higher mutation rate, which will thus prove our hypothesis to be correct, that a missense mutation in the MSH2 gene will actually result in a higher chance of developing HNPCC, specifically because these patients can have an MSH2 mutation gene. Next slide. These are our references, and thank you so much for listening.